In this lecture, we'll be exploring a few of the biotech applications, besides genetically modified plants and animals. The technique that opened up all these applications of DNA technology is called PCR. PCR stands for polymerase chain reaction. This makes many copies of DNA without using living cells. This enables scientists to take a small bit of DNA and amplify it over multiple times. So one gene can be copied over and over again, allowing to give scientists enough DNA to do their experiments. One of the applications of biotechnology is comparing DNA, whether it be comparing DNA of individuals within the same species or comparing different species. When you compare DNA, you're looking for markers. Markers are segments of DNA that are variable among individuals, and they're used to track recessive diseases. Also, you can look at DNA fingerprints, using the markers to make a distinct fingerprint of the genome. To compare DNA, you use a process called gel electrophoresis. So what happens in gel electrophoresis is you take DNA that you want to compare, and you cut them up with specific restriction enzymes. The restriction enzyme cuts the DNA. You put the DNA into what are called wells, little holes in the gel. Then what you do is you run the gel through electric current. Since DNA is partially negative, you run it from negative to positive, and the DNA will migrate to the positive end. The shorter fragments will move quicker through the gel, and the longer fragments will stay far behind. So the shorter fragments move faster through the gel and will travel farther. You can then use these banding patterns of DNA to compare the different samples of DNA to see how similar or how different they are, or maybe they're the exact same DNAs in forensics when trying to solve crimes.